epidemics and even pandemics are occurring at an accelerating pace, and we anticipate to see outbreaks every year or two. So our laboratory has been orienting toward a rapid discovery program where we can respond immediately and seek antibody treatments or preventions in real time as they happen. We've been prototyping this in previous epidemics. We've been preparing for this moment, and now we're activated around responding to the coronavirus. Hi, I'm James Crow. I'm director of the Vanderbilt Vaccine Center.
Dr. Crow is part of a special team of experts, universities, and advanced genetic sequencing groups that are trained to deliver rapid treatments in the midst of an outbreak. It was a concept launched back in 2017 by the Defense Advanced Research Projects Agency.
DARPA usually puts out calls for research about things that can't be done right now. They pitch a vision for something that is desirable. It's an aspirational goal, sort of crazy ideas. So the Pandemic Prevention Program, or P3, the idea was an epidemic occurs, and in 60 days, groups would take blood samples and be ready to treat people 60 days later. Normally, drug programs take a decade or certainly years. So it was a crazy idea. And last year, we did a simulation in which we pulled the time frame down to about 78 days. So we were pretty excited about that success. And it's the fastest anyone has gone from a blood sample to an antibody cure that we know of. And we were just simulating last year, but this year we're doing it for real.
The goal is to safely develop an antibody therapy that can provide immediate immunity, potentially before a vaccine is ready. The clock started to tick once this coronavirus outbreak switched from epidemic to global pandemic.
The pandemic started in China, and certainly we knew in December that something unusual was going on there, but it wasn't clear in December 2019 that this would spread anywhere else. By January, the first case occurred in the United States, and by then, our team got concerned that we really needed to activate around this. Fortunately, as soon as the outbreak occurred, the sequences of the virus were published. It's an RNA virus, and we know that there's only one major protein on the surface of the cell, and it sort of looks like spikes, so people have called it the spike protein. And we know that's what attaches to your cells and attacks you and inserts the virus in. Your body has an immune system, and it responds to threats in various ways. The principal way after you're infected that you prevent the infection from occurring again is through a network of proteins in your body called antibodies. Once you've seen a virus like coronavirus or flu, you remember that virus. When it comes back, you already have antibodies, and they're sort of a shield. So the idea is if we have antibodies that can attach to the spike protein and cover the virus, it'll block the virus from being able to get in. So we know what we're looking for. We want antibodies to that spike protein.
To find antibodies, they need to secure blood samples from patients first.
We actually contacted the providers, the medical teams who are taking care of the first cases. There's a cloud of sort of chaos around these cases. There are family members and press and lots of people in PPE. It's actually hard to get the communication to the subject themselves or their families. When we do that, almost invariably, the people say, I would love to contribute. I would not want anyone else to go through what I went through. These are important samples. We've been working with FedEx Critical Operations, and they've been essential to our success. So not only just barcoding like they normally do, we can watch. The package goes in the truck, and it's coming toward us, and we go, it'll be here in 30 seconds. We do whatever it takes to move this stuff around. But I mean, we do it safely, but we're tracking everything very carefully. We start with whole blood. We've got a lot of red blood cells which carry the oxygen, and the white blood cells. And so we pull the white blood cells out, and those are the immune system cells. And there are the B cells for antibodies. And so we pull those out. It's like a needle in a haystack. We know a few of those in there are for the recent infection, like coronavirus. And so that's where the high technology comes in, is drilling down and finding the individual cells. Now we have a little cell, and it's very fragile, very small, and we have to manipulate that and get it to make the antibody and confirm, yes, I'm a coronavirus cell, at which point we bust the cell open, and inside that are the genes for the antibody. So we recover the genes from a single cell, and we just sequence it, and we can print the gene out in a DNA printer. The machine gives us the DNA back. It's pretty amazing. And that's come out of a revolution in science called synthetic genomics. So you can synthesize DNA at very high scale. And then we can put that back into a cell in the laboratory and that cell becomes a factory to make that antibody. And then we have lots of the antibody, which is a protein we can give to people and protect them. So there's a lot of steps involved, and some of it's sort of conventional, but some of it's sort of magical. If we run into obstacles, we have to overcome those obstacles immediately, like within minutes sometimes. We have about 4,000 or 5,000 antibodies in our pipeline now that we've gotten from single cells from survivors, and then we'll down select. We'll look at about 20 of them in mice, and then we'll end up with 2 of them in monkeys, and we'll be done. That's the sprint. The first human trials of any antibody are likely to be in the June to August window, and that's when we just put antibody into 20 people and make sure nothing bad happens. And then there's a next step where we need to figure out what's the dose. So usually we give 100 people a dose, another 100 people a lower dose, and then finally you do what's called a phase 3 trial. You figure out if it works, and that's thousands of people. And if the antibody works, and we already know it's safe, then it would be released to the public. That's going to take an uncertain amount of time, probably year and a half before it would fully be released to the public, because we have to do this methodically. And you could say, well, you have it, just give it to me. Why don't you just give it to me? But some treatments make people worse. Hopefully by public health measures, social distancing and that sort of thing, we slow things down. It'll remain to be seen whether this becomes a winter thing and we have a whole nother season. So that's why everybody's pushing. It's interesting to watch our team operating because they're kind of in a flow state. These guys are working about 20 hours a day since January, but at the same time, they're actually doing the work at an unprecedented pace for something that's killing thousands of people. So there's a passion and a mission in it. Scientists typically are very methodical. They like to repeat things. They like to be sure. They don't like to tell people the result till they've done it more than once. And in this type of science or discovery, we can't do that. And we're constantly trying to retrain ourselves to think more like, what's good enough? What's the 80% solution? We got to move forward. And it's not natural to think that way.
Coordinated response teams that can deliver rapid therapies will be a critical component in future pandemic preparedness. But how can we get ahead of this next time?
You cannot pick the next one. You can't predict. I know it's going to be a filovirus or a flavivirus or an alphavirus, but one of them's coming. And what we've been advocating is why don't we get antibody cures for all these teed up ahead of time? And when it happens, we don't have this scramble. So we've been methodically making antibodies, and we're about 40 into it. And we have antibodies to other coronaviruses, but they don't cross-react with this one. But I think preparing ahead of time makes more sense than always waiting.